ancient lipids to synthetic life. And what we're really interested in is understanding design principles of simple microorganisms. So since the talk today is kind of oriented, my impression was, for junior scientists, and I put in a few more bit at the beginning with a broader perspective. So I don't know how the timing will work out. I've never tried this before, so let's see. Okay, so to start off with sort of the big question, and then we'll get to the specific thing that we're interested in. One of the big questions is, what is life and how does it work? And historically, people have been thinking about this problem for a very long time. And one of the ways you can think about it is life a machine that is made of parts, and you just need enough complexity in order to replicate a thing that behaves like a living system. And so, I mean, one of the ideas and hundreds of years ago was this idea of these automatons, robots that are like really complex, watches. And if you make one of these things, you can wind it up and it'll walk around and have lifelike behavior. And so, before we had any idea of chemistry and physics and all this sort of stuff, there are a lot of questions surrounding, well, is life like an automaton, or is there some essence, a vital essence that is needed to explain lifelike behavior? So there are a number of experiments that historically made advances in our understanding of this. One was Antoine Lavoisier, and the question he was asking was, can you explain life simply by thermodynamics? Or is there something extra that we don't understand? And so one of the experiments he did is he put a guinea pig or something in a calorimeter, and then he put a block of burning coal in another calorimeter, and he measured the output of CO2 and heat. And it turns out the ratio of those two is exactly the same for a respirating animal and a block of coal. So that was one of the pieces of evidence that suggested that, in fact, thermodynamics can account for living motion. So this ruled out the vital essences that vitalists were proposing. And then later, questions started to center around, what is heredity? Why do cows make cows and pigs make pigs? What's the basis for this information transfer and this fidelity? And so again, there were some crazy notions about vital essences and such things. And one of the model organisms that was explored were frogs, because they copulate for a very long period of time, so they're easy to monitor. And there was an Italian priest named Lazzaro Spalzani, and he was working with these things, and they'd identified that the sperm was sort of one of the key factors in the propagation of eggs. And so he did an experiment where he said, okay, is this the basis for the sperm creating the organism physical and chemical, or is there again some vital thing beyond what we can understand? So he took the sperm from the male frog, flamed it on a fire, and then applied it to the female to see if eggs would be produced. Unfortunately, he messed up the experiment, and the female frog made eggs, and there was about 100 years of confusion until the sterilization studies in Paris years later. So that shows that sometimes you have to be cautious of questioning assumptions in science. Okay, but why am I talking about all this stuff? The point is, now there's an area of there's a field of synthetic biology where one of the questions is, can we build life? And so we're kind of back again to the same problems that people have been facing for hundreds, if not more years. And the problem is, okay, so you always think you know a lot when you've made some progress and learned a lot of things, but what you then end up realizing sometimes is you're back to the same old questions people have been asking for hundreds of years. So the problem is this, right? In the last 100 years, we've worked out the molecular basis for heredity, the genetic code, DNA. We understand a lot about how that is translated into parts, proteins. And we have a good idea for how many of those parts interact in metabolic pathways. But what no one has managed to do yet is to take all these parts, put them in a tube, and have a living self-replicating system come out, right? And so, again, we're back to this problem. Is it just a matter of parts, or is there some vital essence, right? I mean, and here we are in the 2000s. So we have components, but we don't have life. What's the missing thing? I'm going to take you now to an analogy from a cartoon, South Park. I don't know if any of you watched this. But there was an episode where the gnomes came to South Park, and they were taking everyone's underpants. And it was very confusing. And the children chased them down, and they asked them, what are you doing? What's going on here? And they said, it's simple. We have a business model. Phase 1, collect underpants. Phase 2, phase 3, profit. Okay, so in some respects, sin biology is suffering from a similar problem. We have phase 1, collect parts. Phase 2, question mark. Phase 3, build synthetic cells. So one of the key things now is to understand what is phase 2. What are we missing that would allow the ability to assemble a synthetic cell? So what would allow us to explain how life emerged from prebiotic parts before biochemistry emerged? How is signaling coordinated in space and time in cells? And how is development orchestrated in higher organisms? So what is missing to allow us to understand this? One of the things that's missing is the understanding of how life organizes its parts in space and time. And this is a very multifaceted problem. And the little part of that problem that we're interested in is membranes, because in cells, membranes are a key element of cellular organization. And we know that the cell is a homeostatic microenvironment with an inside and an outside the environment. And it has to protect itself from physical and chemical stresses at the same time as remaining open to interacting with its environment and other cells. So you have life and no life, and at the boundary of that, you have a responsive interface. And that's the thing we're trying to get at, is to understand what are the minimal set of parts and principles required to assemble a responsive interface that can support life. Okay, so I stole this slide from Ilya because I like it a lot. It's a beautiful image. You can see at the molecular level, the membrane is a very complex thing. It's made up of a bilayer of lipids and a lot of proteins that all have various functions in the cell. And for these proteins to function properly and interact with each other properly, the membrane has to achieve the right physical properties to support that. So there's a lot of dynamics that have to be carefully coordinated. On the other hand, if you go at the macromolecular scale and you take a membrane, and this is a nice video that Ilya made, it's a giant unilamellar vesicle, and it's about to be stretched from one end with optical tweezers. And the point of this is you see how incredibly elastic this thing is. So not only is the membrane able to maintain these intricate, coordinated molecular interactions, but it's also incredibly mechanically robust. And so what the cell has to do at the fundamental level, before you worry about all of these higher order functions, the challenge the cell has to meet is to create a membrane that can balance all these interactions in the face of environmental perturbations while maintaining its mechanical robustness. And so that's sort of the first order problem that we're interested in understanding how it works. So in our lab, what we're doing is we take several approaches. One is we use bacterial model organisms to study how membranes work. And then we try to extrapolate things from that and reconstitute them in bottom-up synthetic systems. So the basic problem that we're trying to understand is how do we bridge the gap between a synthetic lipid vesicle and something that's robust and is responsive, can respond to environmental changes by adjusting itself homeostatically, and ultimately something that can support growth and division, but that's a long way off. So I'll start with the first problem, which is robustness. We know in eukaryotic membranes, sterols like cholesterol are crucial for membrane mechanical robustness. So why is that? We got a lot of good introductions yesterday to this concept, but in a pure lipid membrane, you can have three distinct phases. There's a liquid disordered phase that phospholipids make. There's a gel phase that saturated lipids form. And then there's a liquid ordered phase, which is the result of specifically of sterols interacting with saturated lipids. And the thing is, as you go from left to right, these become more ordered and less fluid. And the problem is, in a cell, if you go from this phospholipid and you start removing double bonds, which reduces the order, increases the order, eventually you'll end up with a gel phase, and that's where life stops. But when you add cholesterol to that, you end up with this liquid ordered phase, which is more mechanically robust than this fluid phase, but still fluid enough to support life. And so this is one of the principles that cells use to assemble their plasma membrane to make sure that it can resist chemical and physical stresses. Another phenomenon that emerges from the unique behavior of sterols, which we're all familiar with now, is the idea that you can have liquid-liquid demixing of phases. So if you have the right mixture of sterol, phospholipid, and a sphingolipid, some sort of saturated lipid, you can get a coexisting liquid-ordered and disordered phase. And we've seen a lot of images like this where you have, microscopically, you can image these phases coexisting. So here there's the ordered phase is where the dye is excluded. And this is sort of the underlying mechanism, at least in eukaryotic systems and also now potentially in bacterial systems, that could explain how you would have lateral membrane organization for sorting of things to organize stuff. Okay, so clearly cholesterol is very important, especially for membrane mechanically robustness. But then there's a problem, and this is the problem that I got into for my postdoc originally. Cholesterol requires molecular oxygen for its biosynthesis, but we know if you go back in the record of Earth history that there wasn't always oxygen. Back 2.7 billion years ago, before then, there was no oxygen. So what did life do before sterols? And what does life currently do without sterols, like bacteria? Okay, so one of the things that's come up in the literature are the Hopanoids. This is a molecule that was discovered originally in oils. They're abundant, they're everywhere, in almost all sediments where geochemists have looked. And the question was, what are they? They look like sterols. Eventually they were discovered in bacteria. And so people made assumptions that they might be sterol-like. But this was fairly in the air. Okay. The thing about the Hopanoids is they have a very similar pathway to sterols. They derive from squalene and the isoprenoid pathway. However, the unique thing is they don't require molecular oxygen for their synthesis, and it's a much shorter pathway. One step from squalene to diplopterol versus like almost 30 steps to cholesterol from squalene. So these have been found in ancient Earth sediments before oxygen. They don't require oxygen, so they're a good candidate for something that could have predated sterols. They're widespread in bacteria. This is a map of sequenced genomes back in 2010, and the red bar shows you the species that have the Hopanoid pathway. So it's not everything has them, but a lot of things do. And they look like sterols. So if you compare the two side by side geometrically and structurally, they have a lot of similarities. So this question is, are bacterial sterol surrogates, is what got me interested in coming to Dresden originally. So I was a geochemist studying these things in ancient sediments, and I wanted to get more into understanding what they do in membranes. And I came to Kai's lab, and Kai Simmons was an MPI director in Dresden, and he was known for doing a lot of work in the lipid Raft field with cholesterol. And I came and I was trying to convince him to let me come and study the bacterial Hopanoids in his lab. And he wasn't so sure, but eventually he agreed. He said, okay, you can come. And so I went out specifically to ask this question. Can we replace sterol with a Hopanoid and achieve a liquid-ordered phase in liquid-liquid demixing? Okay, so this was the first idea that I came with. It's a very simple experiment in model membrane systems. So the idea, again, going back to this thing, a liquid-ordered phase in the eukaryotic systems is usually thought of as being comprised of a cholesterol and a sphingolipid, or some sort of saturated glycolipid. So we did a few experiments. We've had an introduction to lardin already. This is one of the dyes we would use to monitor membrane lipid order in these model membranes. Again, it's a dye where the emission spectrum is sensitive to lipid packing. And so we can get a ratio, the general polarization index, and this tells us semi-quantitatively something about lipid order. Okay, so the first experiments I did were very simple. Make a GUV with sphingomyelin cholesterol, and then here we have membrane order as determined by this lardin parameter. So high is orange, low order is blue. So we have a liquid-ordered membrane on the left, and we have a phospholipid liquid disordered membrane on the right. And then I replace the cholesterol with a Hopanoid, diplopterol, and we get a liquid-ordered membrane. And we also confirm this with fluorescence correlation spectroscopy that there's fluidity. It's very similar to the cholesterol sphingomyelin mixture. So the next question was, can we get liquid-liquid demixing? So this is a canonical phase-separated GUV with cholesterol, phosphatidylcholine, and sphingolipid, and it's phase-separated with an ordered and disordered domain. And then if we replace the cholesterol with Hopanoid, well, it doesn't do it so well, but it does it. So you get a liquid-liquid demixing to ordered and disordered phases. So that answered the first part of the question, which is, to some extent, in these in vitro systems, Hopanoids can act as sterol surrogates in the sense that they can form a liquid-ordered phase in a pure lipid system, and they can promote liquid-liquid demixing.
When you did this, it's a big difference compared to if you think about the structure of the two molecules. I think it's amazing that they behave. So it is true that it makes I was expecting totally the same feature. You know what I mean?
Yeah. Well, yeah, there are a lot of differences. We could discuss that. I mean, the chain, the flexible hydrocarbon chain in the cholesterol, the extent of the ring structure, and there are differences. One of the things I don't talk about here because I don't have time, but if we look at the interaction of Hopanoids with unsaturated lipids, there's no interaction. There's no ordering effect, whereas cholesterol orders those lipids. So it's clearly not as good in terms of interacting with everything, but in terms of interacting with saturated lipids, it's on par. So, but yes, you're right. There are definitely differences, and you would expect that because they're not the same molecule. So the next question I was trying to get at was, okay, so we know that to some extent there's this analogy biophysically between sterols and Hopanoids, but what does that mean for cells? What are they using them for? What properties does a Hopanoid impart to membranes, and what physiological processes rely on those properties? And specifically, we also have to think about the question, what do Hopanoids interact with in the membrane in order to impart those properties? We know that in the eukaryotic systems, we think of cholesterol interacting with sphingolipids, but bacteria, with a few exceptions, don't make sphingolipids. So which lipids do these things interact with in bacteria to achieve more ordered membranes or whatever they're doing? Okay, so I chose Methylobacterium as a model system, mainly because I had a friend who had made a knockout of the Hopanoid gene, and it was easy to start with that. But they're also very easy to grow. They're relatively simple, the complex life cycle, very robust, easy to do genetics on. So they're a plant-associated bacterium. Every plant has a Methylobacterium on its leaf surface. And they make carotenoids too, which is another interesting story. Okay, so we started with this Hopanoid knockout where the squalene hopine cyclase gene is deleted. They don't make Hopanoids. If you look at a growth curve, the mutant grows about half as fast as the wild type. So there's definitely a growth phenotype there. Now, the first thing was to just purify inner and outer membrane. It's a gram-negative bacteria. And ask which membrane is the Hopanoid or the Hopanoids enriched in. And we did that. We ran here's a nice TL thin layer chromatograph. And we see that the Hopanoids are in the outer membrane fraction, quite clearly enriched. So that answered the first question. They're in the outer membrane. Cool. So then we asked the question, do Hopanoids affect the order of the outer membrane? So we took wild type and then Hopanoid mutant cells and purified the outer membranes, added lardin again, and measured the GP index. And there was a statistically significant reduction in the order of the mutant outer membrane. So that was interesting. We could induce that chemically by depleting Hopanoids from the wild type with cyclodextrin chemically, or loading the diplopterol Hopanoid back into the mutant membrane with cyclodextrin, and we got a similar effect by adding cholesterol in. So that seemed pretty solid that Hopanoids are having an effect on outer membrane lipid order. The next question was, what are they interacting with to achieve that ordering effect? So if we look at the bacterial outer membrane, the canonical idea is that it's an asymmetrical membrane in which the inner leaflet is predominantly phospholipids, and the outer leaflet is made up of lipopolysaccharide, which is this very bulky lipid containing a long sugar chain attached to this very large hydrocarbon component called Lipid A. Now, if you look at Lipid A closely, and you compare it with sphingomyelin, which we'll do in a second, you can see that there are actually a number of interesting chemical similarities between the two. You have amide linkages, and these NHs. You have hydroxylations. Both of those are found in sphingolipids. And these contribute to stronger hydrogen bonding between the lipids, which raises their melting temperature. They also have completely saturated acyl chains, which means they'll form gel at physiological conditions. So that was interesting. And then I asked, okay, is there an analogy between the interaction of a Hopanoid with a Lipid A and cholesterol with sphingomyelin? And okay, I won't explain entirely the experimental setup here, but what we did was we measured the free energy of mixing of a Hopanoid with Lipid A or cholesterol with sphingomyelin using a monolayer setup. And it's a very precise way of measuring the interaction of two lipid species. And what we found was we get the same negative free energy of mixing for the diplopterol with Lipid A and cholesterol with Lipid A, and the sphingomyelin with cholesterol. So there's a thermodynamic analogy between the interaction of a Hopanoid with bacterial Lipid A and cholesterol with sphingomyelin, which is really interesting. And it kind of led us to this currently unproven, untested model, which is that, okay, in the eukaryotes, we have some idea that there is an asymmetric plasma membrane in which the cholesterol and saturated lipids form a more ordered outer leaflet, like Ilya discussed yesterday. In the bacteria, then, the idea is that the diplopterol and Hopanoids could be interacting with Lipid A in the outer leaflet to form a liquid-ordered membrane, where the inner leaflet is more fluid. Okay, so then convergent, but the interesting thing is there's a convergent molecular strategy for ordering the surface membranes between bacteria and eukaryotes. So that's cool that they solved the same problem with related tools evolutionarily. Okay, then the last bit on this before I move on to the next thing is what physiological processes rely on the properties that Hopanoids impart to the outer membrane. There was a lot of work done by many people. Diane Newman has done a lot of really nice work on this, looking at knockouts, Hopanoid knockouts in different bacteria, and many of the observations are the same. When you knock out Hopanoids, the mutant becomes sensitive to pH changes, to detergents, to a lot of things. And so the general interpretation is that indicates that the membrane is somehow losing an important component for mechanical robustness, which makes sense, and that therefore the cells are more sensitive to perturbations. But what I was interested in is figuring out if there's something a bit more nuanced about that, if it was something more than just the mechanical rigidity, but maybe there are actual active processes in the cell that rely on a certain lipid order in the outer membrane. So we did all the standard growth assays, and the one that really jumped out was an assay where we tested how sensitive the mutant was to detergent. We used a non-ionic detergent called Triton X100, and this is a spot assay. For those of you who've never seen a spot assay, what you're looking at is on each row here is starting from very low detergent, like homeopathically low concentrations, up to 10%. And then there's the wild type and mutant, left and right, and each spot is a 10x dilution. So we take the cells, we expose them to detergent for an hour, rinse them, and then spot them out and serial dilute them by 10x. And as you get to the most dilute spot, you start to see colonies. And if there's a lethal effect of our treatment, the most dilute spots will disappear because some fraction of the cells have been killed. Okay, so what we saw was not entirely unexpected. The mutant appeared to have more sensitivity to detergent. There was a lethal effect of the detergent in the mutant more than in the wild type. But the thing that was really exceptional about it, that jumped out, was how incredibly sensitive the mutant was to detergent. We're looking at a lethal effect at 0.01% Triton X100, which is right around the critical micelle concentration of that detergent. So it doesn't seem like something as robust as an outer membrane, even without Hopanoids, would be so radically dissolved by such a low concentration of detergent. I mean, it's possible, but it opened up the possibility that something else was working there. Maybe the cells were just accumulating this stuff and getting poisoned. So a friend of mine that was then in Frankfurt, Robert Ernst, looked at that, and he had done his PhD, master's thesis on multi-drug transporters and said, that's a multi-drug transporter phenotype. So I said, okay, we should test that. So we did an assay for testing multi-drug, ATP-dependent multi-drug transporters in bacteria, and it's a really simple assay. You take your cell, you deplete its ATP sources with some inhibitor, like we use CCCP, and then you take a dye that stains DNA, and when it stains the DNA, its fluorescence increases. So you add it to the cell, it goes into the cell, binds the DNA, and the fluorescence goes up. And then you can use the fluorescence intensity as an indication of uptake and efflux across the cell membrane. So we do this in the wild type, and we let it equilibrate. It gets to a steady state, and then at some point we add back an energy source, some sugar. They make ATP, they turn on the pumps, and they pump out the dye, and you see that nicely. So that indicates that the pumps are active. When we did it in the mutant, not so much. It still does, it's doing something, but it's clearly not doing it at the rate that you see in the wild type. So it looks like one of the phenotypes here is that the multi-drug efflux pumps are defective when we remove Hopanoids. For whatever reason, and there could be many explanations for that, whether it's the channel in the outer membrane isn't gated properly, maybe the proteins aren't inserted properly. There's many things. We're currently in the process of exploring that in more detail and trying to get at the link between Hopanoids, lipid ordering, and the activity of these multi-drug efflux pumps. But it's also interesting because it suggests that, I think as a number of people in this room have also found, if you target certain lipid pathways, it acts as an Achilles heel to bacterial multi-drug resistance. In other words, you can find some way of limiting their ability to pump these things out or resist them, and then apply old antibiotics. So that's potentially an interesting direction to go in eventually. But now for something, what? Yeah.
Is the difference in uptake meaningful there? Just that the curve is higher than the mutant?
Oh, this bump there, we have no idea what that is. It's just like when we...
The fact that the red line is higher to start with and
Oh.
stays higher.
Yeah, sure, because when you equilibrate them, they're already just taking more dye in the first place.
Yeah.
So you already see higher dye levels even when they're active, like growing.
Permeability too.
Permeability, but actually, so it requires about 1,000-fold change in permeability to explain deficiency in this pump assay. I asked a few people about this that do this stuff. So we have maybe a 2-fold change in permeability in the outer membrane when we remove Hopanoids. It's not enough to explain this level of suppression. Yeah?
How about the inner membrane then? Can you rule out that are you sure that the inner membrane is fully energized under these conditions?
That's a good question.
That would also affect that.
Sure, yeah, I mean, it could be a more indirect effect like that, like the periplasm is somehow messed up from the outer membrane being defective, and then the ACR-AB complex in the system. Yeah. So exactly. So we have, I don't think we can make any clear conclusions about what the mechanism is here, but clearly it could be some indirect effect like membrane potential in the inner membrane. Yeah.
So, how does it compare with the LPS mutant or, outer membrane transporter mutations in terms of detergent sensitivity?
I see.
Is it really unusual because this is like an organism I don't know, so is it like really unusual as a sensitivity, or you actually recapitulate something that you get with the LPS?
Well, in this organism, no one's done those studies. So actually, it's one of the things I want to get at eventually is knocking out those genes to make smooth, rough mutants of LPS. But it's a good question. Yeah, I mean, that glycocalyx, whatever, has an important role in preventing these things from coming in. But yeah, there's still a lot of work to do here, but it's an interesting lead. And okay, so now on to the future. So that's what I did mostly for my postdoc, and it's one of the angles that we're following up in the lab. It's our sort of core starting point. But now let's get back to this problem again. So Hopanoids seem to be one of the solutions for robustness in simple systems without sterols. But then the question of responsiveness. What makes a membrane responsive? Meaning, how does the membrane sense changes in the environment and then adjust itself homeostatically to maintain optimal physical conditions? Okay, so what we're looking at here is now the problem is, let's say we have membrane fluidity on this vertical scale, and it's inversely correlated with membrane rigidity. In life, all living membranes try to constrain their viscosity within a relatively narrow range in order to keep the system, the environment optimal for all of these different machines that are trying to operate. So if the membrane becomes too fluid, of course, the barrier fails, it falls apart, and the cell dies. If the membrane becomes too solid, you no longer have diffusion and life stops. So this is really important, and life has evolved to maintain membranes within some window, which is unclear, and one of the things we want to understand, what is that range? And in addition to that, it also has ways of actually maintaining that range in response to perturbations, external and physiological. So if you have a membrane here, and there's maybe a channel or some protein, and then temperature decreases, the membrane will become more compressed, and then you might see the loss of function of a protein because the lateral tension and the viscosity has changed. So then what the cell does is it senses that, and it changes the lipid composition in the membrane in order to restore the original optimal conditions that were important for its function. So this has been known for decades, this idea of homeoviscous adaptation. It's not a new thing. But what is surprising is how little is known about how that's carried out. What are the sensors, and what are the mechanisms involved, and what's the, what would you say, the control system involved in achieving this, right? So that's one of the questions we want to start thinking about in the future. So, okay, one of the things we started doing is, of course, lipidomic. So we can look at the output, which is you perturb the cells and they adjust their lipidome. And that's one of the important things. So, when you do lipidomics, you take your cells, you extract the lipids, you put them on a mass spec, you get a bunch of mass spectra. There's a computer program that filters all that and spits out concentrations of lipid species, put shortly. So we do that, and we started with Methylobacterium, because that's the thing we had around at the beginning. And in Methylobacterium, there's a number of different things we measure, lipids we measure. So we have phospholipids and the Hopanoids. So these are the major Hopanoids. There's some other polar Hopanoids that we haven't yet measured, but they represent a fairly small fraction, and we'll get to those later. So we have diplopterol and a methylated derivative. And then for the phospholipids, there's 4 different head groups. We have PE, PC, PG, and cardiolipin. Chain lengths, we see some variation. It's total chain length ranges between C32 and C36, and these are variants of fatty acids with C16 and C18. And then we see chain saturation. They could have any number of double bonds per lipid from 1 to 4. And when we now tabulate all of these structures, these lipid features that we can measure together, okay, so we did that. We have about 27 species of lipids that we measure. And then we took the cells and we exposed them to a range of varying conditions, like temperature, growth stages, salt concentration. Methanol is an alternate carbon source. They can grow on methanol instead of sugar. And then expose them to detergents, just to see what happens to the lipidome if we just throw all this stuff at them. Okay, so here's the lipidome now. I showed you the lipid features. This is all the actual species, and it's probably a little hard to read, but you don't have to read it anyway. It's organized by head group PG, PE, PC, cardiolipin, and then the Hopanoids down here. And then each of those lines is a specific species with a chain length and a number of double bonds per lipid. And the main point to get from this picture is it's not that complicated. 27 lipids compared with yeast, which have like 200, and mammalian cells, which have hundreds. Right? So that's already a pretty simple lipidome. And you can see already that what I'm showing here, all those dots are the abundances for all the conditions. So you're just looking at like all the data on one. It's a big table, basically. And you can see there's some variation. Lipids go up and down. The red dot is the abundance at the standard growth condition, 30 degrees, pH 7. Okay, so there's quite a bit of variation. If we now ask the question, how much variation and what conditions does the lipidome vary the most over, one of the ways we can get at that is by just taking the standard deviation of each lipid species over a range of conditions and summing that up, and then saying, okay, how much lipid variability was there for temperature or this condition or that condition? So we can plot that. That's what I've plotted here, and what we see is that temperature clearly has the largest effect on lipid variance, and followed by growth stage and then salt, and then the others are sort of in the background. Okay, so that's interesting, but not surprising. Temperature has a huge effect. The other thing is not so much. If we look at now principal component analysis of the lipidomic data, what that does is it's a nice visual way to see how all these conditions relate to each other. What it's doing is simply just showing spatially how the signature of the lipid abundances according to two principal components that fit the data. So what you can see from that is, for example, temperature clearly, again, agrees with this analysis that temperature has a very large effect, and it spreads away from the standard condition, which is in red. On the other hand, most of the other conditions all clump around the standard condition. There's not a lot of, they're all sort of in the same area, so there's not that much change in the lipidome, except for high salt and when the cells go into stationary growth. So when they go into stationary, they do a bunch of stuff and change everything. And those are in a totally different direction from the temperature, which indicates that there are different lipid features being adjusted. Okay, so that's interesting. Then if we look now at how much do individual species vary over all these conditions. So here what we're looking at is the top is a plot of the average abundance of all the lipid species sorted from highest to lowest. And the little gray bar is not a standard deviation error bar. It's just showing you the range of abundances over all the conditions for each of those lipids. So you can right away see that high abundance lipids seem to have a lot of variation. Most of the low abundance lipids don't vary that much. They're really in the background, except for a few in there. And then the heat map down here is, again, the lipid variation is calculated by the standard deviation of a particular lipid species over a range of conditions. And so you can, for each different condition that we tried out, and the main point you get from this is most of the lipids on this map are doing nothing. They're just staying there. There's only a few lipids that are really varying a lot for each condition. So then the question is, which are the lipids that are varying the most, and how many of them are there? So we calculated that. This is a figure that's kind of it's called, what the hell is it called? It's a way of representing a Venn diagram with more than 3 overlaps. Upset plot is what it's called. And so each of these bars, and we're not going to get into detail, just don't worry about it too much, but the point is each of these bars represents a different overlap from different conditions. So the one on the left here shows how many, shows the overlap for all the conditions. And what are we looking at is we asked how many lipids account for more than 90% of the variance for each condition. And so that's here, the set size. So for temperature, there are 13 lipids that account for most of the variability. And for growth stage, it's only 11 lipids. But basically for all the conditions, there's somewhere between 11 and 13 lipids that are accounting for most of the variation in the lipidome. If we now look at how many of those lipids are shared between all of the conditions, in other words, how many are universally changing, that's this left bar, there's 8 of them. So more than half of the most variable lipids are all changing in the same conditions, in different conditions, sorry. Okay, so that already suggests that we have a simple lipidome, but it's even simpler than simple because when you expose it to perturbations, there's a small subset of those lipids that are actually doing anything. So this provides a few hints about kind of what the minimal set of components is you'd need to make a membrane that can adjust to these different sort of perturbations. At the low end, maybe 11. But then now let's look at the actual change in abundance of lipids over temperature. So we go from 4 degrees up to 30. And I'm only plotting here a line plot of the most variable lipids, not everything. And of the most variable lipids, I don't know if you can see it so well, but basically, there's only about 4 or 5 of those variable lipids that are really doing much. So, I mean, if you look at absolute change. So now we're down to like 4 or 5 or 6 lipids out of 27 that are really doing most of the work to help this organism adjust to temperature change. So, okay, there's a lot more we could say about this data set. I'm writing it up now. You can look it up when it gets published. I didn't want to go into great detail, but the main point that I wanted to draw now is just say it's incredible how simple the response is. Even in an already simple organism, there's not so many lipids you need in order to make a membrane that can adjust the temperature range, even a broad temperature range like 4 to 30. So that's pretty cool. Okay, so now we've moved on from Methylobacterium, almost done, and 2 minutes. And so we started working, we wanted to go to a simpler system because 27 lipids, lots of genes, it's already complicated enough. So we identified mycoplasma in the minimal cell as a nice system, and I'll get to that in a second. But the question we really want to get at is how do we understand what the control mechanism is? What are the perturbations? What's sensing it? What's in between? And what's the response? So that's where the mycoplasma come in. This is a nice EM image we got a few weeks ago. Mycoplasma is some of the simplest bacteria that you can grow in culture. The minimal cell that the Venter Institute made has around 500 genes. Other mycoplasma, like mycoides that we're using, have 1,000 or more. So it's pretty simple. They have one membrane, like all our favorite bacterial model organisms. and no cell wall. So it's like a liposome with cytoplasm, basically. And one of the nice things about them for us, biochemically, is they don't make their own lipids. They take up what you feed them in the media. So you can adjust their lipidome just by changing media chemistry without doing any genetics. So that's pretty cool. So what are we doing now? We have, for lipidomic studies, we have a bioreactor system that allows us to make a continuous culture, turbidostat, and to ramp the cells through different conditions, sample them, and then do lipidomic analysis. So that's one of the approaches we're taking. We just sent a bunch of samples out for lipidomic analysis. So next time you can hear about what happens with that. Then we also have been trying to automate a lot of the measurements for membrane fluidity. So we do a lot of measurements in a plate reader using dyes like lardin and DPH. And so now what we can do is we can grow the cells, and we can get lots of growth rates for lots of conditions very quickly, like a tenth of the time it took us before with flasks. And then we can automate measurements of membrane viscosity so that we can now go through a lot of conditions, but we could also potentially then do a genome-wide screen to understand what are some of the genes involved in membrane responsiveness. So, okay, I'll end on this slide. This is sort of some preliminary data that my student Natasha generated over the summer. We just measured DPH anisotropy of Mycoplasma mycoides over a range of temperatures. And we got this. And it was pretty cool because maybe it's not surprising, but there's kind of a range of temperatures over which there's not too much change in viscosity. And then outside of that range, viscosity goes up or goes down. So what this sort of indicates is there's this range where, and outside of that range, the cells die. They don't grow above or below those temperatures. So there's kind of a range here of viscosity where the cell lives. And within that range is a range where they adapt well. And then outside of that, there's, where's the death? Death. Outside of that. So now this is the beginning. We've gotten the system working, and we're really going to start exploring specifically how the membrane can adapt, what is the lipidomic response, and eventually what are the components involved in that response. How does it assemble its membrane? Okay, that's it. There's my wonderful lab who's made it possible to do all this stuff. And if anyone's looking to come to Dresden for anything like a postdoc, come talk to me someday. Yeah, and we have funding from various sources. And thanks very much for your attention.